Hello! We would like to introduce our STH-PAS, a genetic test developed by TBA in Japan. STH-PAS is very simple and easy test for anyone and can be used anywhere. An overview of the STH-PAS test, the procedure, and the materials needed. For PCR reaction, it needs a primer set. For our STH-PAS test, it needs a tag primer and biotin primer. Tag is a short DNA sequence. For target DNA or RNA of the sample, suitable primer sequence is designed. As an example, there are 4 kinds of primer sets with 4 different tags and biotin primers. After PCR amplification, the target-specific amplicon is obtained. The amplicon has a tag on one side and biotin on the other side. Then add the development solution and streptavidin conjugated blue latex beads. The blue latex beads bind to the biotin. Finally, a PAS strip is inserted into the tube. The amplicon flows horizontally from bottom to top by the developing solution. On the PAS strip, TBA has already printed complementary DNA in line from bottom to top. 1, 2, 3, 4 for each tag. 1, 2, 3, 4. Due to hybridization, tag of the amplicon is captured and accumulated on the detection line. This gathered amplicons show blue color by latex blue beads. This is the basic principle of STH-PAS. Total cost of reagents and equipment required for STH-PAS is from $1,000 to $3,000. Compared to other methods, it is very reasonable. The introduction of STH-PAS will contribute to the prospect of great cost reduction. No special knowledge and no expensive equipment is required. STH-PAS is an extremely simple genetic test using an inexpensive equipment. However, the quality of the test results of the experiments can be trusted. We have already provided our STH-PAS to many researchers at research institutes, hospitals, and universities around the world. The advantage of STH-PAS is high sensitivity. Visual detection technology is generally considered to be less sensitive than fluorescence detection. However, the STH method uses a primer with a single-stranded tag, and this part remains single-stranded even after PCR, so no denaturation step is required. In addition, there are no competing complementary strands, and there is an opportunity to detect all PCR products during chromatographic detection. This results in high sensitivity, which is comparable to the fluorescence method. The second advantage is the easy addition of multiple detection. Our DNA chromatography can detect multiple targets simply by adding a detection probe. The third advantage is that strips can be used in common. Even if you have a number of detection groups, you can use the strips in common by using a common tag for target-specific primers within the group. These are 3 advantage points of STH-PAS technology. The Dengue subtype detection kit is designed for detection of dengue virus subtype genomes in plasma. The kit contains a primer mixture, developing solution, latex bead solution, and nucleic acid chromatography strips. After RT-PCR, you can visually determine the existence of the target gene using the DNA chromatography method. The Dengue subtypes, Type 1, Type 2, Type 3, and Type 4 are detected from bottom to top on tags 1, 2, 3, 4. Due to hybridization, the kit is easy to use without requiring a specific instrument except a PCR machine. We compared our Dengue subtype kit with QPCR methods using a dengue-positive clinical specimen. This is the result of a comparison of our Dengue subtype kit with the CDC's kit. Our Dengue subtype kit showed a high correlation with the CDC's kit. Therefore, STH-PAS sensitivity is as good as QPCR. The viral RNA must be extracted from the sample before using this kit. The recommended method is to use a commercially available spin column method. This method can extract a pure viral RNA and detect it with high sensitivity. However, it requires a high-speed microcentrifuge and many steps for extraction. Recently, we have developed a new kit that can easily extract using a Serum Direct extraction reagent. The Serum Direct extraction reagent is simply mixing with serum and heating at 75 degrees Celsius for 5 minutes. Comparing the spin column method and the direct method is inexpensive, simple, and quick. However, the sensitivity is slightly reduced. Now we will introduce the Serum Direct method. First, add 16 microliters of Serum Direct extraction reagents to PCR tubes. Add 4 microliters of sample serum. Flash the vortex and flash the centrifuge PCR tube. Heat the PCR tube 75 degrees Celsius for 5 minutes. After heating, store the tubes on ice until the next step. PCR reagents are prepared on ice according to the recipe in the table. Then add 2 microliters of the extracted RNA solution. Flash the vortex and flash the centrifuge PCR tubes. Store the tubes on ice until set in the thermal cycler. Set the tube to the thermal cycler and start the program following condition. It is strongly recommended to perform the reaction with a hot start. Heat the thermal cycler to 52 degrees Celsius in advance, and then set the ice cooling reaction tube. Mix the dilution buffer and the latex beads, 10 microliters plus 1.5 microliters per test, with the vortex mixer. Open the caps of the PCR tubes after RT-PCR and add 11.5 microliters of latex developing solution prepared before. Mix the tubes thoroughly with the vortex mixer and flash centrifuge. Insert DNA chromatography strip into the tube and wait 10 minutes until blue lines appear. Be sure to hold the absorbing pad region of the DNA chromatography strip with dry hands. Leave them at room temperature for 10 minutes for developing and judge the results. Perform the reaction at room temperature 20 to 37 degrees Celsius and over 40% humidity. A low temperature and humidity may result in an insufficient reaction. Thank you for watching our STH-PAS introduction video. If you have any questions, please contact TBA. More information about STH-PAS is available in the following technology introduction video.